What I want to do with this talk today is to really give some insights on how to optimize oligonucleotide separations and give you some insights into the column technologies and why they've actually been made. Now, when we look at an oligonucleotide, you've got various flavors. So you've got the double-stranded DNA, you've got single-stranded DNA, you've got that version in the ribonucleic acid versions, and you have the big plasmids and vectors that we're using. Now, these vary quite a bit in size, and that changes what you need to do with the column, or maybe it's even which column you use. Now, the oligonucleotide separation, just even within the last year, the interest in that's dramatically increased, and that's partly because of the new drug-based products that are oligonucleotide based. We've got things like the short interfering RNA, which block the overproduction of some proteins that happen in disease states. We've got the messenger RNA vaccine therapies, and we've also got gene therapy itself, which is replacing a defective gene. And usually that disease has no known other cure. So there's a lot of new technologies coming out where we're using oligonucleotides. Now, as you put an oligonucleotide into the body, it's nice if you've got a drug and it stays around long enough for it to work. Now, the body's full of nucleases. These will just destroy any oligonucleotide that gets introduced, especially a foreign one. Now, what they've done to make these more stable and resistant to nucleotides, a common modification is just to replace an oxygen with a sulfur in the backbone, and that makes the molecule stable. Now, we'll go through how we can look at that and ensure that's been done properly. Another common impurity or a common analysis is to, if you've synthesized or synthetically made some RNA or DNA, you like to know it's the right length and the right sequence. And it's quite, we can lose a base pair or even lose two, and the loss of that base pair can happen anywhere in that sequence. So you've also changed the sequence. So we want to make sure we've got the right amount or a good amount of the main product and very little impurities. And of course, we'll talk about some other impurities as well that we can look at. This is by no means an extensive list of what can happen with these molecules, but it gives you an idea of some of the most common ones and where we can do to affect separations. Now, I'm going to talk about two different types of analysis, and I'm going to run through these relatively quickly because I want to get through more of the points of how you do this separation with anion exchange and ion pair reverse phase. And really, I'd say even now, anion exchange has got that bit better separation power, but the reverse phase, it's now compatible with MS. Now, one of the things I'd like to say up front, it's an ion pair reverse phase using amine ion pairs. Those amine ion pairs will contaminate the LC, the tubin, anything that goes into the mass spec, your transfer capillary. So usually these systems are dedicated to this analysis. Now, I'll show you how you can get around that in a little way as well in the further separations. But we'll move straight into anion exchange. We've got a selection of columns. I'll show you why they're made and which ones to go for, all the way up to some prep work. Now, a lot of ion exchange columns are out for protein separations as well. Now, not all anion exchange columns are the same, and the ones we're using for oligonucleotides are specifically different for specific reasons. Most common anion or ion exchanges for proteins will have a solid core resin, and you'll have the ion exchange sites grafted with a long polymer chain to present it to a protein. That's not how we do oligonucleotides. Oligonucleotides, the resin structure has changed. We've still got the solid particle, which is highly negatively charged. Now, that would repel the negatively charged oligonucleotide, but we've put micro latex beads all the way around that, which are positively charged. Now, that increases the surface area, so it's increasing your capacity. You actually get a lot better resolution from this type of resin than you will from a grafted resin. So it's a specific resin for oligonucleotides. So don't go thinking you can just use any anion exchange columns. These columns are specifically made for oligonucleotides. And the first one out was a 13 micron resin. I'll go through that. And the newer one's a smaller particle size. Now, what I want to introduce in this talk is how DNA analysis has been done in the past. And the gold standard column for DNA analysis or RNA analysis was always the DNA pack PA100. Now, that's a 20-year-old column. This column used to be called the Nucleopack column. It's the same column, it underwent a name change. But this is an old column brought out before Thermo Fisher Scientific had even acquired Dynex. So it was a Dynex column at the time. The new columns are much better. They've got better pH stability, much better temperature stability, and I'm going to show you some slides where you can use pH and temperature to change your separation, and the resolution's better. So there's no reason to be buying DNA packs just because you see it in SOP or a lot of publications. You go for the DNA pack 200. The other thing I'd like to add, to achieve that position of the gold standard columns, we've never advertised this. This has gone through word of mouth. That's how good these separations are on these columns. Now, this is an idea of a standard separation of a synthetic product, which is quite key in QC, where you've got the main product and you're looking to see how many failure sequences and how many impurities have I got in there. Now, that separation, it's actually size-based. You've got the biggest thing coming out at the end, then smaller things in front. And you're saying, come on, Ken, this is an ion exchange separation. How can it be size-based? Now, when you look at it, you've got a DNA molecule, and every time you add an extra nucleotide base, you're adding an extra negative charge. So as the chain gets longer, it also gets more charged. So even though the mechanism may be ion exchange, the separation is actually based on size. And I'll show you how to affect the separation on size and push it to a purely size-based separation, and how you want to separate same length oligos on sequence variations as well. Now, I'll push on with the sized. Now, this is an example of the two columns that I'd like you to look at more strongly. This is the DNA pack PA200, which is really your workhorse column. And here we've got a 25 centimeter long column, and that's separating N+1 from, even at the level where you've got a 54 base pair long molecule, separating it from a 55. That's just one base pair difference. And that's an easy separation for this column. But you can see the 34 and the 35 that's an easier separation, and it's a wider separation. And you look at the one below, and you've got the high resolution RS column, and you think, well, resolution's not that much better. But if you look at the time, that's done in 15 minutes. The high resolution one does it in about 9 minutes. This is another example I'd like to give you of a difference between the two columns. This is an example of poly-T. Now, poly-T is it's a good separation to give you an idea of how good the resolution is. And here on the DNA pack PA200 with a 25 centimeter long column, we've now got 69 peaks separated in 15 minutes. Now, if you look very closely, there's smaller peaks underneath around the 5 minute area. Now, these are the same length chain, but it's an isomer. So it's either a linkage isomer, but you're now separating same length isomers on this column. And we're separating that same length sequence because we're using the most powerful eluent for this system, which is sodium chloride. I'll go into the use of perchlorate later on, but if we used a perchlorate system, you wouldn't see that isomeric separation at the bottom here. So that's the standard column. If we now look at the RS high resolution column, again, we've done the separation in 10 minutes here, but you've now got 83 peaks. Now, that's based really on the fact that you've increased the resolution of these low abundant isomeric peaks between about 1 minute and 5 minutes here. So you've got it's a very, very high resolving column, this. So you've got the choice of the two. Now, I'm now going to go on and show how you can affect the separation on these columns. Now, with a gradient curve, that's the same as any other HPLC column. You can optimize your gradient and optimize the separation. What I'd like to spend more time on is how you can change the selectivity on these columns. And one of the key motivators on this is the eluent salt. You can really change the eluent salt and change the selectivity. And we can see what solvent, temperature, and pH is actually going to do as well. But one thing when we do an analysis, especially with large molecules, we've got to look at not just what the column's doing and the interactions on the column, what's the molecule itself doing? What condition is that big molecule in? And if you're using poly-T, fine. If you start using poly-G is very well known for aggregation. It'll stick to each other. It's got complementary binding that'll just bind together. And if you look at the panel A on the left here, it's a mess. Everything's just come out in one big lump. Now, this is exactly the same sample that's been loaded on panel B, and you can affect that separation in two ways. You can increase the pH just by adding a little amount of sodium hydroxide, and that will keep those chains apart and allow the separation, allow the column to do its work. Same thing gets done if you increase the temperature. And I normally do these sort of runs at about 70 degrees C just to avoid any problems I'm going to get that could be complementary strands, complementary sequences, which might get these molecules to bind together and cause problems in the separation. Now, we'll go and look a bit further at temperature. Now, temperature on an ion exchange column for this separation is doing the opposite of what you would expect on a reverse phase column. It's making things bind that little bit stronger. So if you look at the separation at 30 degrees C, and here we're separating a short interfering RNA that's been made up with a synthetic antisense and sense and joined together to form the duplex. And we can separate the duplex from the single-stranded products, but the big thing is we can also separate the same length sense and antisense strands because they'll obviously have a different sequence. They've got the complementary bases on each side. So here we see at 30 degrees C, the separation's not brilliant, but as soon as you get to about 40 degrees C and onwards, the separation gets much better. So you can look through 40, 50, 60, 70, but when you get to 80 degrees C, that's too much. And at 80 degrees C, you've actually melted the duplex and you're forming now the single-stranded sense and antisense strands. So to do this properly, to see how much excess sense or antisense you've got in the formulation, you'd be running it at about 70 degrees C or a little bit less. So the thing we're using here is sodium chloride in the eluent as well. That's to get the best separation and to allow the separation of the same length strands. And you can get the decent separation here. Now, what happens with pH now? Now, if you look at pH, we've got a different trigger to allow separation here. And that trigger comes from the G&T bases. We've got a tautomeric oxygen atom on these particular bases, when what that means is we can actually change that from a non-charged form to an extra negative charge. And that the pK that happens is between pH 7 and 11. So between that pH somewhere for G&T, you're actually going to form an extra negative charge. And of course, that will give it extra retention on the ion exchange column. Now, as you go up, you can see with sodium chloride, the difference between pH 7 and 9 is not much. But as soon as you get from 9 to 10, you've got a big increase in retention. And that's because you've created that negative charge. Going from 10 to 11, you've got that bit more. Going from 11 to 12, it's fully on the negative form there, so it's not making a difference going any higher. Now, when you look at that pH, you're not going to do that in a silica HPLC column. But I will mention this later on. So this trigger will not be available to you if you're actually using a silica column. If you use perchlorate, same thing happens because it's nothing to do with the eluent salt. This is a pH thing. So the same jump you're getting between 9 and 10 and 10 and 11. But if you look at the salt concentration, you're using about a fifth of the salt concentration with that chaotrophic ion, that chaotrophic salts perchlorate. So it's much less salt, and even then the retention time's pushed in. So that's what you get. And the other one you'll see, if you look carefully, perchlorate pushes the separation to more size-based. It stops that same sequence separation or reduces it considerably. If you look at the pH 11 trace in orange, you can see the sodium chloride has much more impurities in the baseline here than you have with the perchlorate eluent. It's mainly just that size-based separation. So here, if we go on now and we'll look at the effect of solvent. And again, solvent does the same sort of thing. If you increase the solvent concentration from none to 20%, it pulls the separation in, it reduces retention. Now, if you've got a hydrophobic molecule on there, such as in this case, we haven't removed fully the tritol protection group. It's a hydrophobic molecule, and that's going to increase the effect you'll find with acetonitrile. But basically, it does show you that there is some hydrophobic interaction on this column if that separation's moving in. And you'll see that in even more detail here, where we look at molecules where we've specifically added a group that is hydrophobic. So biotin and a lot of the fluorescent derivatives, they add a hydrophobic group, and you can see the separation with sodium chloride here. You've got a wide separation, the peak's getting broad. If you add acetonitrile, these peaks get much sharper. So if you have got one of these hydrophobic groups on there and you want same sequence separations, you should really add some acetonitrile. But even when you add the acetonitrile, it does move the separation across to size only as well. And again, if you use perchlorate doesn't really require the acetonitrile with these hydrophobic groups. It disturbs that interaction already. So perchlorate's another way of using these hydrophobic molecules. I'm now just going to go through some odd linkage position errors you can get. Now, normally you've got a 5' to 3' linkage all the way through the chain, but you can get these aberrant linkages where it goes 2'5" instead of 3'5", and that will change the structure of the molecule. So you're physically changing the way that molecule's bent. And because of that change in structure, it'll change the retention on the ion exchange column. And here with the separation here, you can see in the middle, the one marked 0 is a pure product, all 5', 3'. The other products are where we've changed that 2', 5' linkage and put it in different places along the length of that chain. And we're separating all the way through there. So that's a clean separation. So the other thing you've got, which is an important one, and I've shown this slide for two reasons. One is that we've already said, if you're going to use this as a drug product, you need to protect that from nucleases. So you will change that oxygen to a sulfate group. Now, when you do that, you're doing that right on top of an asymmetric phosphate group. So you've now created two possibilities on whether that sulfate goes on one side or the other. So you've now got a distereoisomer. Now, even that distereoisomer, it will separate on the ion exchange. So you can see the non-modified product in black at the bottom in the chromatogram. If you add a thiol group there, it's changing. You've now separated the two distereoisomers in blue. If you put that in a different position, again, you get a different separation. If you actually put both of those on the molecule, you've now got 4 possible distereoisomers, and now it separates all 4. Now you think, oh, that's great separation, fantastic, and that's a sodium chloride. Now what I want you to think about is that's a 37 we're looking at here. Now normally you'll fully thiolate that molecule, and when you've got 37 sulfurs on that, your number of distereoisomer positions is immense. You'll never ever get a column capable of separating that many distereoisomers. So what tends to happen, you get a broad humpy peak where the column's trying to effect their separation, but it won't manage it. So your peak shape looks horrible with sodium chloride. That's where you now need to go to a dumb down the separation and use perchlorate and force the separation to size only. So for this separation on a fully thiolated molecule, you'd always go to perchlorate. The other thing about this, as you notice this separation, as you add an extra sulfur, it gets more and more retained. When you've got them fully thiolate, it sticks like glue to that column. It's a very, very strong retention. And that retention actually helps us. Because if you want to look at what your new drug, you've got a short interfering RNA, got a lot of sulfurs on there, sticking to the column like glue, and you want now to measure that in liver extracts, how much of it's in the liver, how much of it's in the kidneys, how much of it's in the bloodstream, you can now just do a homogenization of that liver, spin down the debris and inject. All the proteins will come out a lot earlier than that certain RNA. Even the large messenger RNA comes out in front of it. And here you've got a beautiful separation of the product you want to measure right at the end. So I haven't done any extraction here. It's just worked in one injection, and it's linear. And you can even see some of the breakdown products or the impurity products here as the N-1, 2, and 3. So that's a good way you can actually look at clinical studies. Now, this also works when you're looking at cell lysates, where you're trying to make or look at supercoiled large DNA. And in this case, we've just taken the cleared lysate and injected it all. And you can see the protein, the small RNAs coming out in front, the large RNAs in front still, but because your main product that you want to look at, the supercoiled large plasmid, it's coming out way at the end. And here we're separating the supercoiled, and in this particular sample, there happens just to be a lot of relaxed linear separation in there as well. So you've made a linear supercoiled analysis there, and that was this is an old slide. This was done on the PA100. I'd really like to see this done on one of the newer columns, which should be a lot better. So if anyone out there's got some samples that'd like to try on this, I'm very keen to have a look at this and update that slide. The other one I've said, you can't use ion exchange from MS. Well, you actually can, and it's got a couple of benefits. We can do the separation, nice separation. You can actually fractionate that with a fraction collector, fractionate the main peak, or you can just do a hard cut automatically and then trap onto a reverse phase trap. Here we can use the DNA pack RP, which I'll speak about later on, but we've also done this with the Acclaim PA column, which is a polar embedded resin, and that's just a trap column, but it allows the use of very, very low solvents. That will trap the DNA. Then we're just going to wash this off that trap with 40% methanol. So we're not using any amine ion pair. So that means you can use this on any HPLC and any LSMS system you've got in the world. It's not going to poison the system with the amine ion pairs. So it's a way of getting the DNA into an MS after a separation without using the ion pair. This is an example of how it's done, and we can get this down to 1 minute. But what you've got, you've trapped the DNA onto a solid support, salt rushes straight through. You can really wash the salt off dramatically. You can even put an EDTA wash in there to make sure all the salt's gone, because salt addicts they're not something you really want in an MS trace. They don't give you an advantage. And then all you're doing is washing off with 40% acetonitrile, with 40% methanol, and you can actually reduce the flow rate as you go into the MS. Keep it in there for longer, and then go back for fast equilibration. Now, what's that look like in the MS? This is the raw data on the left, and then you've got the deconvoluted data on your right. And you can see you've got a very, very small amount of a sodium addict in there, but it's clean. And you've got a nice mass evaluation there. And there's new software available now for deconvolution of oligonucleotides. That's commercially available. And I think it's now the first commercially available sequencing software we've got there as well. So software is available. So this sort of analysis and this column technology can be quite useful, especially to laboratories which don't really have the time or the equipment to dedicate something to just purely oligonucleotide analysis. I'm now quickly going to go through the prep columns. Now, this is a different format, and it's quite interesting. For a prep column, all people do normally is we'll just pack the resin into a dirty, great big column. In this case, this is what we've done with the DNA pack PA200. This is a big column. It's 22 millimeters wide, 25 centimeters long, and the capacity is 3 mgs. Now, if you look at the DNA Swift on the other side, it's much, much smaller. It's a fraction of the diameter, and it's shorter, yet the capacity is up at 50 mgs. How did we manage that? Now, really, the column particle for the DNA pack 200, it's a surface exchange resin. They're not designed for high capacity. Where what we've got here, we've used a monolithic support, but we've used exactly the same amide functional latex beads. So you've got the micro latex beads that are on the surface of the DNA pack 200, but here you've got much, much more of them embedded in a porous monolithic support. So you've got a big porous tube in that column, and there's a lot of latex beads inside there, so the capacity's gone up. The back pressure's actually gone down on this column as well. And if you use this, you can see the purple trace on the left corner. That's the analysis of the crude sample we're going to purify, and it's 78% pure. That's the analysis done where we've overloaded a little bit the DNA Swift column, but you've got a purification. You take the peak and then reintroduce that for a further analysis and the same analysis, and you can see that the purity now has gone up to 97%. Now, you can trick the system as well. If you want to, you can way overload the column, and it's called a sample displacement technique. This has gone well past the peak capacity of the column, but still within the total column capacity. And your peak shape here looks horrible. But the advantage you've got here is really the thing you want to purify is the last thing coming off. You've got rid of your earlier impurities in the front, or your N-1, N-2s, and you're collecting the stuff here, the main peak. And when you look at that for reanalysis, it's relatively pure. You've got a 90% pure fraction here. So that's the way to do prep and analysis. So really, oligonucleotide retention and selectivity can be changed. If you want a size-only separation, go to perchlorate. If you want the ultimate separation and same length separations and sequence separations, sodium chloride. If you've got something big and sticky, I would tend to go to perchlorate just to get it off with less salt. But you can change the pH, solvent, and temperature, and you'll get a good separation. And you can get these fractions automatically desalted straight into MS without any really extension on your analytical time. It's only taken a couple of minutes to do the desalt. And you've got some new columns there that are good. I'd like now to go through and spend some time on the reverse phase ion pair work. Now here we've got a column that we've designed again specifically for oligonucleotides. Now I'll give you the advantages of what we've done here. It's not a standard C18 silica column. We've managed to get a color for the collars and the end fittings, which are a fluorescent yellow. So really, you don't really need to look at the label. You pretty much know what column you've got just by looking at it, and you can see it from the other side of the laboratory. So that's what the columns look like on the outside, what's happening on the inside. Now, when you're looking at ion pair reverse phase, you've got a reverse phase particle here. That's hydrophobic. This oligonucleotide is not going to stick to that. That's highly charged. So again, we're going to introduce an ion pair. Now, normally when we're looking at small molecules, an ion pair will bind to the small molecule, make it a neutral molecule, and then that neutral molecule now binds to the resin. Here it's a bit different. We're using a lot of ion pair. The amount of ion pair reagent we're putting in there will totally coat that resin surface. So what you've done effectively is converted your reverse phase resin into a pseudo ion exchange column. And that's why these separations look very similar to anion exchange. You've got several ion pair reagents that you can use. I'm not going to go through them all. I tend to find the most commonly used is one of the best ones you'll find, which is your triethyl ammonium acetate. We have got differences where we use LCMS. We don't want that much ion pair in there. So you can reduce the ion pair concentration for LCMS by adding a reagent called hexafluoroisopropanol, which I'll name as HVIP for short. And that's what's usually used in LCMS. Now, we can do the same sort of separations, but I want to go through quickly, what do you want out of a column that's going to do these big, long oligonucleotide molecules? First thing, a small, smart particle size column is not going to work. They're not going to get in the pores properly. You'll not get a good separation. And here we've gone from an 80 pore size resin, which is on the top left, to the one on the bottom left, where it's 150. And this is just using a 19 to a 24-mer. And you can see you need that extra pore size to make a difference. And the resin structure used here is exactly the same. The only difference is the pore size. And now, but when you go to an even bigger molecule, we're now looking at restriction endonuclease fragments. And look at the 150 pore size one here. It's not good enough anymore. You need to go to a 400 at least to get that separation you need now. Now, the other problem with this, you think, I'll just use the 400 angstrom pore size. However, when you go to that 400 angstrom pore size and use it for the smaller molecules, the separation with 150 now looks better than the 400. So now you're looking at, I need 2 columns to do all these molecules. Luckily, you don't. You just need the one. The DNA packed reverse phase column we've got now will resolve both small and very large oligonucleotides. And I'll show you that and prove that in some of the slides going forward. Now, there's your column here. Now, this column is not silica. It's the same polystyrene you've got in your ion exchange columns. So it's now a polystyrene column. And where I said before you couldn't use pH to affect that separation, now you can, because that polystyrene resin's not going to dissolve. Do a high pH run at pH 10 to 12 in a silica column, and within a few runs, you've got an empty column and a lot of dissolved silica. This one is now going to be stable at pH and at temperature. Now, the pore size is variable. It's super macroporous. So it's huge. These pore sizes are big. It goes up to a 200 angstrom. But because it's super macroporous and has a varied pore size all the way through, it will separate the small ones and the big oligonucleotides in one column. And there's a new column that's come out for this, which is 25 centimeters long, just to give you that extra separation. Now, I'm not going to dwell on this slide because there's a lot of ion pairs. What I will mention is that The triethyl ammonium acetate here, I would regard that as equivalent to your sodium chloride and ion exchange. That's your super element. That's the one that's going to give you the most separation, the one that's going to give you the same length separation. But if you want to move up into push the separation to pure size based, that's when you'll go to the bigger ion pairs such as tetrabutyl ammonium, or here we've got hexyl ammonium acetate. That's the sort of thing you'll use. So it's the same sort of thing. We're just using a different ion pair to push that separation from sequence based in size to more size based. And with ion pair reverse phase, you can push it further across to pure size based than you can with ion exchange. And if you use LCMS, you're just going to drop that ion pair concentration with HFIP. Now, what's these separations look like with the similar separations we showed on ion exchange? Now, here's 8 minutes. So you've got that same separation, but now we've done it in 8 minutes. So this separation's good. What I will say is that you've lost now the isomeric separation in between these strands. So you've lost a little bit of separation power, but that's 60 peaks in 8 minutes. That's powerful. And what's it look like in comparison in more detail? That's in blue, we've got the ion pair reverse phase. Bottom black, we've got ion exchange. You can see the peaks are slightly sharper in the ion exchange, but the resolution's pretty much the same, and the selectivity is different. You'll notice some of these peaks that are coming up in the ion pair reverse phase aren't really the same as the ones that's in the ion exchange. So you have got a selectivity difference, so it's worth looking at both. Now we're going to go through the same sort of format. I'm not going to spend too much time on gradients because that's relatively obvious how you optimize a gradient. The big difference is I'm always using a pre-column heater. That's because reverse phase applications are more sensitive to temperature than ion exchange. Make sure you're at the right temperature right at the start by using a good pre-column heater. And I'd prefer an active pre-column heater rather than a passive one. But we're going to look at what pH and ion pair you can use and some additives to selectively change the separation. Now here it's the same sort of thing we've shown before with ion exchange. And here you've got method development. This is the effect of a pre-column heater. If you don't use one, your separation's not going to be as good because I'm normally doing these separations at 70 degrees C. Get an active pre-column heater. And so that's what the panel on the left's shown. The ones in the middle here are the effective gradient shape. And again, it's the same sort of thing. If you're using a polymeric separation or you want to separate a lot of N+1s, the earlier separation is easier to do than the one at the back. So you will flatten the gradient out at the back and speed it up at the front with a curved gradient. They can be quite powerful. But in normal circumstances, if you've got a synthetic product or mainly one length, you'll optimize a linear gradient along the area where your oligonucleotide will elute. And this is the effective temperature. And it's exactly what you'd expect. As you increase the temperature, you lose retention. It gets nearer and nearer in. Now, what I want to show you now is what this column's mainly going to get used for. If you're using this column, most people are going to use it with an LCMS system. And most of these systems will be dedicated to this analysis. Now, at the top left here, you've got the tick. And here you can see the main product is the main peak with an N-1. And here you've got the UV trace here. Now, you can get a very good quantitation here of the N-1 with the UV. But one you've got on the mass spec, that N-1's a different molecular weight. And the other thing about that N-1, it could be any base, and it's usually all of them, because you could have lost that base on any length of that sequence, so it could be a different base, or it would be a different base you've lost through the sequence. And here we're seeing through the identification with MS, it has lost all 4 of those bases. So that N-1's not just a single molecule, it's a set of variants containing different missed bases, or where you've missed one base. And there's the TIC of the main one looking different at the top. Now, this is a separation which is particularly difficult, and it's where LCMS can come into its own. Now, you can separate N-1 and PO separations from, say, a fully thiolated Cerner. You can do that on ion exchange, but if it's hit and miss whether you can do it or not. It's very sequence dependent. Some molecules will allow that, some you just won't manage. And it's the same with the ion pair reverse phase separation. Here we're looking at different extremes of temperatures, and we can see that N-1 in PO separation, it's better done at between 80 and 90, you're getting good peak shape. But here we've got the red one, which is an N-1, the blue one's a PO, where you haven't fully thiolated, you've lost an S, and the black one's the full product or the good product. And you're separating them both, so you are separating the N-1 from the PO. But you can use EDTA as an additive, and EDTA has been known to sharpen the peak shape. I'm not saying it's going to work in every instance because I've seen it not make any difference. And the other thing with EDTA as an additive is, if you look at the top trace, that still does what you want it to do. It separates the N-1 from the main product. So it's up to you whether you try EDTA or not. And this is an example where we have used EDTA, but it gives you an example of a full-length product in black and how you can separate the different POs and the different N-1s. But the more you've got of these impurities, the more chance you've got one will overlap on top of the other one. But what we're looking at here when you've got MS, that phosphothioate is a different mass to the product. You've changed the sulfur from an oxygen. Same if you've got an N-1, you've lost a base, it's a different mass. So here where you've got the tick on the top and the UV at the bottom, you can identify these two and quantitate from that UV trace there. But in reality, if you've got an N-1 on top of the PO, you can't. You're then going to have to go to extract the reline chromatograph to do true quantitation on this. And here's an example of methylation, where we're looking at methylation here as well. And again, you've added a methyl group to that to the cytosine here, changed the mass, and the mass spec's going to see it. But you will affect the separation here. So if you've got a UV separation, great, you've got quantitation. But if you've got multiple things overlapping on top of each other, you're now going to have to go into the MS data, do an extracted ion chromatogram, and quantitate the N-1 separately from the PO from anything else that's in there, methylation. And how accurate can this be? And when you've got this, here's an Orbitrap where you've got a Q exactive, and here they've really thrown the resolution up quite high. And but in this case, you're looking at a major peak in the tick at the top left here, and you're looking at a very minor peak here where you've got the N-1. But if you look at the extracted ion chromatography, the extracted ion mass on that N-1 is beautiful. It's got beautiful sensitivity, and that's easily quantifiable. And the mass accuracy is phenomenal at that stage. For such a small peak, you've got 0.4 ppm error on that. And even on the main product, which is the 35-mer, you're down at 0.1, but your mass accuracy is good, and your sensitivity is good, and your quantitation will be good. So you can do these sort of things. So it's one of the things in techniques LCMS does come into its own, where you've got multiple things overlapping in the same peak. So here you've got controls where we're looking now at the bigger things, and how you can look at a ladder that's been generated by just using a restriction endonuclease map, and you've got different molecular weight base pairs here. So you've got different amount of bases on there. We can now look at a PCR product. And again, this is using the ion pair in DNA pack RP, but we've just thrown the whole sample on there. So this is the lysate, the cleared lysate. So the PCR products are coming out early on. So this is where you've got your building blocks. They're the small building blocks for the purification or the synthesis. They're coming out early on. You've got your product, which is in this case it's a 500 base product. You can see it's coming out in the right place for a 500 base product when you overlay it with the control or your map at the top. And if you've got other impurities where this differs from ion exchange, proteins stick harder. So the proteins will come out in the wash. And this is, and again, it's another example where you've got a vector. In this case, that vector's, the double-stranded vector's been cut to release the DNA that you actually want. And the DNA has actually been unzipped to produce single-stranded DNA. So you can see the two 500 long DNA strands there separated as well. So that's an example of how you can look at what you've actually got in your plasmid and how you can actually make sure what you want is actually in there. Now, just to go through this in terms of a summary, if you've got ion pair reverse phase at the top, if you want size-dependent separations, in some cases that is important, especially when you're looking at things like cerners, where you really want to concentrate on the size-based impurities. You don't want to try to separate distereoisomers. All that's going to do is give you broad, horrible peaks. So we're now using tetrabutyl iron. It's the bigger strong iron pair. It's going to push the separation to size. And if you use additives such as HFIP, that also does that. So again, if you want a sequence-based separation and get the biggest possible separation power you want, just use triethylammonium acetate. No additives, just that sort of iron pair, small iron pair. Now, if you go to ion exchange, you can do exactly the same thing. Push it to size base by throwing a K-tropic ion exchange salt in there. Use perchlorate. Also, if you add some acetonitrile, that'll do the same thing as well. So if you want to do size-based, use the sodium chloride with no additives. Now, can this be useful? What about 2D chromatography? Now, I've already mentioned this N-1 in PO separation is difficult to do. Now, what happens if you actually use the power and use the selectivity on two different columns? Now, the first column dimension here, we're using tributyl ammonium acetate, and the DNA pack RP is an iron pair separation. And we're looking at a cerner. We're looking at it's about a 22-mer short interfering RNA. And we've pushed the separation as far as we can to size-based. So what we've done here, we've done a separation, we've overloaded the column a bit, and we've separated out the N+1 and the N-1, 2, and 3. Now, if we take that fraction there, then rerun it on the DNA pack 200, and now we're going to look for a selectivity on sequence selectivity. We're now pulling out the PO that you deliberately didn't resolve with the iron pair separation in the top. So now you know fine well that peak coming out in front on the ion exchange is a PO because you've already got rid of the N-1s. So that's an example how you can use HPLC on its own in 2D to do that difficult separation. Now, for a summary, you've got a lot of columns, and these aren't just any columns. The DNA pack columns are polymeric columns specifically designed for this purpose. In the ion exchange columns, we're not throwing our protein columns at this. We've specifically designed a column for DNA analysis. And when you look at the two things separately, reverse phase is where you'd normally go for LCMS, but you can get samples from an ion exchange into an MS doing that fractionation or that hard cut. Size and sequence separations, you can tune on both. You can do the thiolated oligonucleotides, but you really need to push to a size-based separation. And the high-resolution MSs now are looking very good. And the software you've got for this, which has just been released, it's much, much better. So it's going to make this arsenal of what you've got for oligonucleotide analysis much, much more powerful. But initially, even if you're using MS, you've got to get that separation into the MS first. And this is how you're doing it. And MS compatibility would need that desalting for the SACS analysis. Now, really, what I'd like to go now is give you an idea where we've gone with this. And on the bottom right's the things I've just discussed, the nucleotide columns. But if you look at the bottom left, we've got the same sort of capabilities for proteins and monoclonal antibodies. Here would increase from one, on the nucleic acids we use anion exchange, proteins will use anion and cation exchange. And again, we're using the grafted columns for this. And the ProSwift column here is there as well. Now you've got the MAB pack columns, which is very similar to the DNA pack column. So that's the same sort of functional group. It's a polymeric column and it's a porous resin. And that's got the blue colors on rather than the yellow. And if you go to the peptide columns, the Acclaim columns and the AccuCore columns are very good. The advantage we've got with those is we've got the AccuCore Vanquish column, which is 25 centimeters long. It's a longer column than you'd normally buy, still a small particle size, but that column length gives you very good peptide separation. The same goes for the Hypersil Gold. The C18 1.9, that comes in a 20 centimeter long column. When you look at glycans, we've got the amide glycan column, which amide helic, which is a normal column you'd use, but the glycan pack columns, they're very important. They're a mixed mode column, and they have anion exchange and reverse phase or anion exchange with helic. and that gives you the extra ion exchange separation alongside the helix of the reverse phase. This is important where you've got really complex glycans that are heavily silated. It'll pull the silations out till you've got one silation, 2, 3, 4, pull them into groups, and then it gives you the different composition inside those groups. So you can get much more separation power on complex glycans with these glycan mixed mode columns. Now I'd like to leave you with that, but before I go, I'd like to give some acknowledgements to the people that do this work. I don't do all this work. There's too much work for any one person. Julia and Shanwar and Chris from Sunnyvale and Jim, they've done a tremendous amount of work preparing these columns, making these columns. No one knows more about making columns than Chris. No one knows more about using them than the other three. And a lot of these slides come from that laboratory. Jennifer's, again, Thermo Fisher, and she's helped set the separation up, and Stacey's the product manager. Now, I thank again VWR for allowing me to do this presentation, and I've had a lot of help from Mark Dickman at Sheffield University in preparing some of these slides, and I've learned a lot from Mark as well. So really, I'd like to open up the presentation now to questions.
Thank you, Ken. Really excellent presentation there. And we can see there's some questions coming in. A reminder for people who may have questions now, please do type them into the questions tab, and we will answer them now in this session for you. So the first question for you, Ken, what is the LOD and the LOQ and the LCMS method approximately?
That's
around slide 40.
Yeah, that's a difficult one. Well, we're looking at I'd say when you're looking at the LODs, if you're looking at the fact that you've got a big main product and you want to look at the impurity levels, the impurity levels we're seeing now are in the grass, so they're beyond what you could probably see with the UV and with the normal programs you use for DNA deconvolution. I would look closely at the newer one. Thermo Fisher have just brought one out, and we're seeing things that you wouldn't even see in the tick sitting in the grass there now. So are well below the 1%, 0.1% level for this of an impurity. But it's not just the column separation and what mass spec you're using. The Orbitraps are sensitive. The software is going to make a difference for that as well. If you're looking at UV, again, you're well below the 1% level and down into the 0.1s for impurities.
Thanks, Ken. Next question is, what would be your suggestion for the separation of 100 mer oligonucleotides with MS compatibility?
I'd say for that one, again, you've got both options. If you really want to have a look at the separation, you'll get some powerful quantitation by UV from impurities from that. And if you want to look at the 100 mer, you can actually desalt into the MS. But certainly if you're going from 100 mer, I wouldn't go past the DNA pack RP. It's a damn good column. And you'll just about start, you're at the level of at 100 mer, struggling to separate N-1, but it's close. But it's one of the best columns I've seen for that. And again, I'd be pushing the triethyl ammonium acetate eluent system with the HFIP.
Next question is, do you have columns for morpholino oligonucleotide analysis?
Again, we've done quite a lot of these. Now, you've got so many modifications. When I originally started doing oligonucleotide separations, I thought, well, that's easy. You know, it's 4 bases in a line, can't go wrong. The number of modifications you've got on this are phenomenal. Now, most of them, there's a way of doing it. Sometimes you can separate them by ion exchange or by UV. But when you've got mass spec and the power of mass spec after it with the DNA pack RP, you can usually see a mass difference. So even if you can't separate, you've got the power of the mass spec behind you. So I'd say, yes, go for it.
Okay. What are your recommendations for purity analysis of long oligonucleotides, such as gRNA and mRNA therapeutics?
No, if they're quite big, you're not going to get an N-1 separation on something that big. It's not going to happen, whatever column you use. There's a couple of options. One, you can actually just run it and try looking at the MS and try with the MS going to as high a resolution as you can and see what you can separate on some of those peaks. You will get a separation of some things, especially with the DNA pack RP. You've seen the 500 base pair separation you had on one of the earlier slides. That'll still be available. But if you've got N-1s, N-2s, you'll not see it, but you can delve into the MS and start looking for that. The other option to look at that is to cut that big thing down into smaller pieces. So actually use restriction engine nucleases, cut the beastie down and look at the smaller ones, either separately or together.
Thank you. So would there be a limit of acetonitrile concentration you'd recommend for the anion exchange methods?
You don't really need to go that high in the solvent concentration. I've never gone higher than 15–20% for the anion exchange. It doesn't seem to make that much difference if you go much higher. The hydrophobic interaction on that column's not that much, and usually once you've got to about 20%, you've really got rid of it. So that's not much point in adding anything extra unless you want to change the molecule itself, or you've got something particularly nasty and hydrophobic on the end of that DNA.
Still lots of questions coming in. So how do you quantify impurities on MS without reference standards?
Again, it's all going to be relative. So you're going to be looking at relative quantitation unless you have the reference standards. So that's nothing you can do about that. You can try having a look at how these things fly. When you've got a protein sequences and peptides fly very differently in the MS. And again, you can get dirty grade big peaks and very small peaks that do not like the MS at all. With the DNA separations, the molecules you've got, whether it's a 20-mer or a 22-mer, it's going to fly in that MS pretty much the same. It's the same sort of molecules, so you haven't got as big a problem with relative quantitation as you have with a protein or with a peptide. But I'd have a look at that and see what the relative separations look like. A lot of this stuff we're doing with MS now, it's coming out and it's relatively new, because a lot of the software is relatively new that's come out. We've never really been able to do sequencing before, and now the sequencing software is now available. You've got people doing MS where they'd normally only do MS, and even getting the fragmentation conditions right for MS, it's pretty rare. It's just not that many people doing it. So I'd say it's a work in progress, but in general, for relative quantitation, it's not as problematic as some molecules.
Next question is, what is your recommendation for separation of oligonucleotides from tissue homogenate? Which column can be used?
I'd go ion exchange, because, and again, it's what molecule you're actually looking at. If you've got something that's big, that's going to stick on the ion exchange column like glue. If you've got a short interferon RNA that's fully thiolated, that's going to stick on like glue as well. Everything else is going to come out in front. Now, the common method for extracting these is to use anion exchange. But if it's coming out right at the end of the separation and you're going to do a separation or a quantitation anyway, just do an analytical run, and the impurities will all come out in front with anion exchange. You can get away with a lot on reverse phase, but remember your protein is going to come out with a different selectivity. The proteins will come out a lot quicker on the anion exchanger. They'll actually bind all the way through with a cell culture on a DNA separation. So the other option for that is to treat the separation with something like pronase or proteinase K, get that protein all the way down and get rid of it, get it into small pieces, and then do that little bit of sample prep before you then go on to ion pair reverse phase. And then you've only got oligonucleotides to deal with.